So good afternoon to all. Thank you so much for the invitation. Probably my talk is really going to be entirely different with what so far you heard. Fundamentally, we are moving towards the basics to application science. So we specifically trying to produce or find the novel source for producing co-sorbates, that's a liquid separating proteins that could be used for basically for biometrical applications, for different something like tissue engineering or basically which focus on wound healing development. So our topic is we basically working on biomaterials. Biomaterials, as you are aware, that those are biologically compatible and it could be introduced into the body because it has a long biological compatible activity. And especially when you talk about the biomaterial, you immediately come to know that collagen, gelatin, or some elastin like proteins. Those are really good because they are biologically compatible, but still it has a lot of limitations. Especially we are much interested on developing some underwater like wet resistant adhesive materials because those kind of materials are frequently available or synthesized only by the chemist people. It has a limited level of biological compatibilities. So we are very much interested on finding and novel wet resistant biomaterials because when we use those kind of materials, the water is a strong repellent which never going to allow you to, it's not going to allow you to adhesive in any position. So we are really searching for some of the highly wet resistant material from marine and other sources. Some of the findings are it has been really reported that some of the marine organisms are generally produce some of the proteins. They are denatured, disordered proteins. They will undergo for a liquid separations that form a fluidic and protein rich condensed co-sorbates, and that hold the wet resistant mechanism, but that also hold the preserve the dispersion of the proteins even in the aquatic environments. So we are very much interested on those kind of proteins and we are looking on it. We try to develop, we identify and we will recombinant express and we will try to understand the mechanism of how these co-sorbates are really occur in the wet resistant conditions. One of the material what we are really working on is like barnacle inspired proteins and also the Mussel inspired proteins. Today I am just going to talk about some of the role and understanding the Mussel food proteins on wet as a wet resistant proteins. So basically, I think comfortable here. Okay. So basically, if you could see these muscles, probably everybody knows that when you walk on the seashore, the muscles are strictly adhered in the seashore, the rocks. It produces some of the proteins called food proteins. Usually it produces the vessels, it produces the thread, and the thread containing n number of food proteins, basically from food proteins 1 and 2, 3 to it's go up to the variety of around 29 proteins. These proteins will go for a liquid separation among them, and it resulted in the condensed co-sorbations. Those co-sorbates usually help for the attachment and it facilitate even they are very highly resistant even the high tidal. So when you look at that specifically, the muscle food protein 3 and 5 form very stable thermodynamically strong co-sorbates. That's look into the people are very much interested on it and they still we exactly know the mechanism. Most of the time it has been proposed that these muscle food proteins are containing TOPA, that's one of the post-translationally modified amino acids from tyrosines play a very critical role on it. Because TOPA is a catapholic amino acids, it's a versatile amino acids. What's happening over there? When it get oxidized, what's happening? It's going to be a topoquinone. So it's highly reactive in nature. So it will always prone to go for a intra-cross linking. But in the case of reducing conditions, it will form a it's dehydrated. It will go for a very interesting hydrogen bonding interactions as well as metal coordination, metal oxide coordination. But as I mentioned, the TOPA oxidation is play a very critical role on wet adhesion. When it's going to become oxidized and it form a topoquinone, it prone to just release from the attached surfaces. So the questions here is how the seashore, sea is, as we are aware, that sea is thermodynamically oxidative conditions. So at these conditions, how these muscle food proteins are really maintaining the strong adhesive nature is still unknown. One of the recent study, one of our collaborators reported that when they introduced this topo molecule on it, and they observed that it go for the intramolecular interaction between the topa that specifically hold the adhesive nature in the water, but still exact mechanism is underexplored. And most of the time when we try to study the muscle food proteins and related to the adhesiveness, all the studies are mainly focused on topa or topa based chemistry. We call that topa paradigm, because based on the theory, lot of topa based material has been proposed, and a lot of interesting adhesive material has been developed. That's very good. But what here is not unexplored is these proteins also have several other amino acids like phosphoserine and hydroxyproline and hydroxyarginine. These proteins also present almost like 15 to 20%, but still it is not explored. Even recently, one of the studies very clearly informed that some of the hydroxyproline that present in the similar kind of co-servate forming protein, slime protein, and they play a very important role in the adhesive interactions. And we hypothesize that because, as you also aware that the hydroxyproline is play a very critical role in the collagen. It maintains the triple helical structure, stability, and also play a very critical role on the intracellular assembly, the fibril formations. So we hypothesize that whether the hydroxyproline may play any role on in the case of co-servation formation by helping hydrogen bond formation, or in the case of dehydrations. But the question here is, why so far people have not tried to explore that one? Because one of the reason could be we don't have a specific methodology to understand or introduce such a kind of post-translational modified amino acids into the protein itself. Even we could introduce site specifically, or even we could introduce only one amino acid, post-translational modified amino acid itself become very challenging, because we have to go for enzymatic reactions to study all the modification, which is very difficult to control. And moreover, in this case, we have to introduce minimum two post-translational modified amino acids to the protein, then only we can suggest something. So here, that's a major challenging and the biggest challenge for us when we started studying such an experiment. So, because why even synthesizing through different approaches may not work here. We need bulk amount of samples to mimic similar kind of study the bulk adhesiveness to propose or understand more about the co-servation mechanisms. So we have in our lab, we developed a new approaches. We are working on this for several years. What we are trying to see here is we introduce unnatural amino acids, that's a post-translational modified amino acids in any amino acids into the protein through either two ways. One is we use genetic code engineering approach, where we try to misaminate the amino acylation there instead of our target amino acid. For example, if I want to introduce proline, we completely deplete the proline in the medium, and we will apply and we will introduce a hydroxyproline in the system. That what will happen, our proline tRNA and tRNA synthetase will recognize, and it will go and introduce through misaminylation. So, and second one is, we are also working on that one, but the first one is the major challenge would be, it's going to be random. I mean, it's going to be residue specific. Wherever the proline will be there, it will be replaced, and we cannot control site specifically. But we also have another method. We develop usually orthogonal tRNA and tRNA synthetase system, which can specifically recognize our unnatural amino acid, and it will be going and introduce site specifically at particular position. For example, if you want to introduce at some of the active sites, we could introduce site specifically by developing orthogonal translation machinery. So in our lab, the first one, what we developed, the first important thing is we overcome the challenge. We develop a single and double unnatural amino acid incorporation. What we did is, like, we selected the one of the genetically modified E. coli, that's the oxotroph system. We have optimized the system to introduce proline, hydroxyproline instead of proline. And we also developed a multi-auto oxotrophic system where we have introduced TOPA and hydroxyproline simultaneously in the systems, and we observed that after that whether the system is really going to form a co-observation or not. We induce a co-observation conditions, and where you could see here very interestingly, when proline alone, we could observe only the aggregation. It's not going to form any kind of co-observate micro droplets. But in the case of hydroxyproline induced or the TOPA, or even the multi unnatural amino acid incorporated co-observates, we could clearly see the micro droplet formation, which is one of the best indicator of the real co-observation, the liquid separation occurs. So we studied them further to understand. We collect the samples and we centrifuge and we collect them co-observates, and it's subjected to the fluorescent analysis. We treated with diafluorine, and you could see here when we introduce two unnatural amino acids, 5 times increase in the fluorescence. It's almost like compared to the other the fluorescent intensity was very high. So it's clearly indicate that when we introduce two unnatural amino acids, it go for a crosstalk and increase the co-observation efficiency. And also when we analyzed with different pH the role, because we just mimic the C conditions, we treated with different pH, and also we treated with different kind of salts, and we could observe that at the level of 6 to 8, the double unnatural amide, I mean when the protein having TOPA and also hydroxyproline, it goes a very good separation over there. When we study the phase diagram, and we could notice that when we have a two unnatural amino acids, I mean TOPA and hydroxyproline, the amount of protein required for co-observation formation is very less. So compared to the TOPA, I mean earlier everybody believed that the TOPA is really play a very important role. But here we could see that the two unnatural amino, I mean hydroxyproline containing co-observates also facilitate in the co-observation formation. But one thing, as I mentioned, TOPA is very good in the case of the, it's really help and facilitating the wet adhesive nature. But the question comes here is, what will happen when the oxidation happen? Whether it's going to completely disperse the co-observate mechanism, all the hydrogen bonding will go off, and it will disperse the co-observation formation. So what we did is, we treated the protein with in the oxidation condition, that's sodium periodic, and we observed that here you could see very clearly when the muscle food protein, where we do not have any post-trans-modified amino acids, it's still very stable because it's not going to form any co-observate. Still aggregates or the aggregations are maintained. But in the case of proline loaded co-observate, still after oxidizing, it retains the micro droplets. But in the case of TOPA containing co-observates, very interestingly, you could see that after once it get oxidized, it's completely lost the co-observation pattern. You could see the micro droplet has disappeared. But in the case of two unnatural amino acids, I mean where the protein encoded with hydroxyproline and TOPA, still it maintains the co-observation efficiency. And in order to make sure that whether it is really to support further, the proteins were we study the electropotential of the proteins by cyclovoltammetry analysis. You could see there before oxidation, the TOPA containing protein containing a good level of electropotential. But after oxidation, you could see here the TOPA containing proteins and the hydroxyproline almost lost its electropotential. But very important things what we have to observe here is the two unnatural amino acid. I mean when the protein having hydroxyproline as well as the TOPA, it retains almost 50% of the potential capacity. So it clearly says that the formation of the hydroxyproline, I mean introduce the hydroxyproline may form the transition peptide bonds, which may facilitate or it may really help to maintain the tautomeric equilibration of TOPA during the oxidation conditions. And we also studied the oxidation potential and also the co-observation formation in the case of bulk adhesive nature, because really it maybe works in a very least amount, but we have to make sure that that kind of bulk adhesiveness is really happening over in the system also. Unfortunately, the video is not running here. So we have observed very clear pattern that when it get oxidized, the the hydroxyproline containing co-sorbates are converted to topoquinone. When it become converted as a quinone, it lost its co-sorbation ability. However, when in the case of proline containing hydroxyproline containing amino acids are loaded with the system, so somewhere it supports it, and especially it facilitates to maintain the original characteristic feature even the tidal oxidative environment. But the question again comes here that that's everything is okay, but the problem is really the hydroxyproline is play a very important role, and can you confirm it again by any other approach? So what we did is we have instead of hydroxyproline, we have introduced the butylproline. That's one of the hydrogen breaker, the hydrogen bond breaker. When we introduce those unnatural amino acids instead of hydroxyproline, and we could clearly see that all the proteins are lost that co-sorbation formation. That's we couldn't observe any kind of micro droplets over there. So it's clearly indicate that our hydroxyproline is really facilitating the formation of co-sorbation at the oxidative conditions. And we also studied the adhesive patterns using AFM, and we could observe a very clear-cut pattern that when we have two unnatural amino acids only, we could now maintain the similar kind of bulk adhesive nature. And our bulk adhesive natures also confirm that when the shear strength of two unnatural amino acid containing proteins only have the ability to significantly shows a higher level of adhesive nature compared to the single one of either with the hydroxyproline or TOPA. So we hypothesize that finally the studies, what we conclude is when the TOPA alone, it could be fine, it could efficiently go and bind with the different surfaces, but when during the oxidative conditions, still it exactly, we almost exactly know that what exact mechanism going on in the surfaces when the plaques are producing different kind of proteins, but we could at the moment conclude that, or some kind of suggestion we could say that the hydroxyproline or other hydroxylated amino acids also could play very important role similar to the TOPA. It could help to maintain the oxidation potential, although it has been reported that some when muscle food proteins also produce or co-segregate with a lot of antioxidants to maintain the oxidative potential in that particular environment, and also similar kind of amino acids or the hydroxyproline, hydroxyarginine to the hydroxylated amino acids also have play a very important role. So we could give a equal priority that such a kind of modification also should be explored. So the ultimate idea is to create what is the use of creating or understanding the co-sorbates in our cases. Like we try to use those kind of co-sorbates to mimic like a extracellular matrix to prepare your organ-like structures like organoids. So those kind of material could be useful to study, so like an animal model. Maybe we try to replace them with like a kind of animal model. So we use this kind of co-sorbate as a ECM matrix compared to collagen other cases. So we explore the possibility of our co-sorbates to prepare different kind of spheroids in our lab. So compared to other naturally using collagen, our co-sorbate also really supports our hypothesis that it wonderfully form a very good spheroids. So these are some of the co-culture pattern, and just show the applications, real applications of our co-sorbates in the formation or different tissue engineering applications. These are some of our publications, and this mainly the work has been done by one of my graduate students, Cecilia, and also now partially doing with Sonam. So especially all the work has been completely done by our lab group member. Thank you so much.
At the end of the talk that I'll give tomorrow afternoon, I'm going to propose that the nature of the hydrogen bonding that exists inside an LLPS condensate is fundamentally different from what exists outside. And the reason that proteins by themselves, under conditions of macromolecular crowding, or DNA, or proteins with DNA, proteins with nucleic acids, the reason that they form LLPS spheres is basically correlated with their ability to hydrogen bond with water. So I find it deeply satisfying that you not only showed that hydroxyproline and dihydroxyphenylalanine, when they are introduced, the ability to form coacervates is increased, but you also said that this might happen with other hydroxyl group containing amino acids also.
Yeah.
This might very well be the case. In fact, I'm also going to plug the same hypothesis tomorrow afternoon.
Okay.
In fact, serine containing, threonine containing proteins with a very high content of these on the surface might actually show a greater tendency to coacervate with lower CSAT values and so on.
Yeah.
So I believe what you're saying, and I think you demonstrated it very nicely by showing that when you form the quinone, you lose the ability. So basically what you're losing is hydrogen bonding with water.
Yes.
You're losing the ability to reorganize the water in the droplet.
Yes.
Yeah.
So that's a major challenging. Whenever we people are trying to use DOPA-based chemistry, the question arises here is what will happen when DOPA get oxidized? Because it has a strong tendency to go for intra-cross linking. And for example, during the reducing condition, it will very well coordinate with metal oxides, and even transistor metal ions. But when it becomes converted as a quinone, so it lost all the ability. And we could observe here also that when it converted as a topoquinone, the coacervate also disposed. So the question again is, what's really happening over there? Okay.
Loss of the ability to
Formation, yes.
Yeah, a fantastic talk. So I was wondering that do you know anything about this? Because there are also a lot of people working with this calcium or metal and coordinated dopamine polymers like Frank Caruso in Australia. And so I've never seen they are actually talking about liquid phase separation. And I don't know whether they really didn't care about the fact.
No, no. The main thing is the interesting fact is this chemistry. What you are talking about is actually the TOPA is really fantastic material. Actually, it will go for a variety of chemical reactions. So people, like as you said, they will use it for calcium, maybe it for a metal coordination. Yes.
Or iron through too, I guess.
Yes, yes.
Too, yeah.
Even frequently people use the topoquinone in the TOPA also, because everybody use, if you look at very carefully, they will control the pH.
Yeah.
But most of the time, any TOPA-based material, when it become reach around pH around 7.8, the question arises here: how is it really going to be stable? Somewhere it's good, somewhere it is problematic. Like we also did some work that we conjugated with some of the drugs, like boronated drugs. It will very well coordinate with it form a boron ester formation. But when it reaches the pH of 7.8, it will dissociate. The drug will be released. So you can also use it as a carrier, and you can use it for a controlled, a pH dependent drug release also.